Hi guys, welcome back. For most of human history, we believed one fundamental thing about inheritance. You pass your DNA to your children, and that DNA is fixed. Your genes are your genes. What your children inherit from you is written in stone at the moment of conception. That belief has been overturned by one of the most significant scientific discoveries of the last 20 years. What you eat, how you manage stress, whether you exercise, what toxins you're exposed to, how much trauma you carry. These aren't just affecting your own health. According to a rapidly growing body of peer-reviewed research, they may be leaving measurable marks on your DNA that your children, and in some cases your grandchildren, could inherit. This field is called epigenetics, and in the next few minutes you're going to discover exactly how this works at the molecular level, the specific lifestyle factors scientists have documented leaving heritable marks on DNA, the evidence that trauma experienced by one generation can appear as epigenetic changes in the next, why a study published in Nature found that a father's diet before conception directly altered the metabolic health of his offspring, and what this research means for choices you're making right now. Let's get into it. First, the most important distinction in all of this: epigenetics is not about changing your DNA sequence. Your actual genetic code, the sequence of base pairs making up your genes, doesn't change based on how you live. What epigenetics describes is something different, and in some ways more remarkable. It describes changes in how your genes are expressed, which genes get switched on, which get switched off, and how strongly without altering the underlying sequence at all. Think of DNA as a book containing every instruction your body could ever need. Epigenetics is the system of annotations, the highlights, the bookmarks, the crossed out passages that determines which parts of that book actually get read and how often. These annotations come in three primary forms: DNA methylation, the addition of chemical methyl groups to specific sites on the DNA molecule, typically silencing or reducing the expression of the genes at those sites; histone modification, changes to the proteins around which DNA is wound, affecting how tightly or loosely genes are packaged, and therefore how accessible they are for transcription; and non-coding RNA regulation, small RNA molecules that control gene expression at a post-transcriptional level. Here's what almost nobody outside of research labs fully appreciates about these mechanisms: they are responsive. They change in response to your environment, your behavior, and your experiences. And in certain circumstances, they are heritable. They can be passed to the next generation through egg and sperm cells in ways that researchers are only beginning to fully map. A 2026 review published in the journal ACS Publications, covering the most recent advances in the field, confirmed that transgenerational epigenetic inheritance, the transmission of environmentally induced epigenetic changes across multiple generations independent of DNA sequence alterations, is now one of the most active and rapidly expanding areas of research in all of biology and medicine. But here's where most people get this completely wrong. When people first hear about epigenetics, they sometimes assume this means DNA is infinitely malleable based on lifestyle, that you can fundamentally rewrite your biology or your children's biology with the right habits. The honest picture is more nuanced than that, and the research makes this clear. Some epigenetic marks are reset between generations through a process called epigenetic reprogramming. Most of the methylation patterns in eggs and sperm are erased and re-established during development. But the research is also showing that this reprogramming is not complete. Some marks escape the reset, and those are the ones that carry information from one generation into the next. The question the science is actively working to answer is exactly which marks, from which exposures, survive in which tissues, and the answers are becoming clearer every year. Let's look at the specific evidence. One of the most frequently cited findings in this field came from research published in Nature, examining what happened when fathers ate a chronically high-fat diet before conception. The study found that the resulting female rat offspring showed beta cell dysfunction, i.e., impaired insulin-secreting cells in the pancreas, even though the offspring themselves were never exposed to the high-fat diet. The epigenetic marks left on the father's sperm by his diet we're influencing the metabolic health of the next generation. This is not a single isolated finding. A 2025 review published in Frontiers in Nutrition, examining the full body of research on lifestyle, diet, exercise, and epigenetic modulation, confirmed that dietary patterns, physical activity, and even mindfulness practices produce measurable changes in DNA methylation, histone modification, and non-coding RNA expression, and called these lifestyle factors crucial roles in modulating epigenetic mechanisms with implications for disease prevention and health across the lifespan. Smoking is perhaps the most well-documented lifestyle exposure in epigenetic inheritance research. Prenatal tobacco smoke exposure has been shown to produce global and gene-specific DNA methylation changes in offspring. Altered methylation in sperm from paternal smoking has been documented in multiple studies. These changes affect gene expression in the next generation in ways associated with increased risk of respiratory disease and other health outcomes. Chronic psychological stress produces some of the most striking epigenetic findings. Research published in Biology and Psychiatry documented that early life stress produces epigenetic changes that are then transmitted to subsequent generations, with offspring showing altered HPA axis function and stress reactivity even when raised in completely stress-free environments. The experience of the parent, not the environment of the offspring, produced measurable changes that persisted across the generational line. This is one of the most sobering findings in epigenetics research. Trauma leaves molecular marks, and those marks, in some documented cases, can appear in the biology of children and even grandchildren who never directly experienced the original event. Research examining the descendants of Holocaust survivors, of famine survivors, of individuals who experienced severe early childhood adversity, all have found measurable epigenetic differences compared to control populations in patterns consistent with inherited epigenetic modification rather than shared environment alone. But here's where this gets genuinely hopeful, and this part is as important as everything else. Epigenetic marks are not permanent in the way genetic mutations are. They are dynamic, they respond to intervention, and the research on positive lifestyle interventions is just as compelling as the research on harmful ones. A 2025 Frontiers in Nutrition review documented that regular aerobic exercise produces consistent positive changes in DNA methylation patterns, particularly in genes associated with inflammation, metabolic regulation, and even tumor suppression. Exercise doesn't just change how your body feels, it changes which genes are actively expressed in your cells. Diet quality has perhaps the most extensively documented epigenetic effects of any lifestyle factor. Specific nutrients serve as the raw materials for epigenetic processes. Folate, vitamin B12, choline, and methionine are all directly involved in DNA methylation chemistry, meaning the food you eat provides the building blocks your epigenetic machinery uses to annotate your genome. A diet chronically deficient in these nutrients doesn't just fail to support your health in general terms. It literally impairs the molecular machinery responsible for appropriate gene regulation. Mediterranean-style dietary patterns have been specifically associated with favorable epigenetic profiles in multiple studies, including methylation patterns linked to reduced cardiovascular risk, reduced inflammatory gene expression, and in some studies, measurable effects on biological aging as measured by epigenetic clocks. Mindfulness and stress reduction practices have also produced documented epigenetic effects. Research has found measurable changes in inflammatory gene methylation patterns in long-term meditators compared to controls. The mechanism appears to involve the reduction of chronic cortisol elevation, which itself drives epigenetic changes in stress response genes. And crucially, many of these positive changes are occurring in tissues that include reproductive cells, meaning the potential for positive epigenetic inheritance runs parallel to the documented evidence for harmful inheritance. Let me bring everything together. The discovery that your lifestyle modifies not just your own biology, but potentially the biological inheritance of your children does not reduce to simple determinism in either direction. Your children's health is not simply a direct download of your habits. Genetics still matters enormously. Epigenetic reprogramming between generations erases many marks. Individual variation is enormous, but the evidence is now clear and published in peer-reviewed journals across multiple decades and multiple research groups. Environmental exposures, dietary patterns, physical activity, chronic stress, trauma, and toxin exposure all leave measurable epigenetic marks. Some of those marks survive the transition between generations, and they influence gene expression and therefore biology and health in the offspring. A 2026 review published by the American Chemical Society confirmed that transgenerational epigenetic inheritance is now considered a cornerstone of modern biomedical research, with clinical applications being actively developed. The choices you make today, what you eat, how you move, how you manage your stress, what you expose yourself to, are operating on a timescale that extends beyond your own life. The real question was never just what kind of life do I want for myself. The real question is, but what are you writing into the biology you will one day pass on? If this changed how you think about your own choices and your future, subscribe, because next week we go even deeper into the science that shapes who we are and who our children will become. See you in the next one.